So, good afternoon, everyone. Welcome to today's seminar. I have two speakers online, which is Karen Miga and Benedict. Benedict, I'm not sure if I'm pronouncing your name correctly. They will be presenting on telomere-to-telomere work that they've been doing over time. So let me just, while Miga is Dr. Karen Miga and Dr. Benedict Paten, they will actually be presenting on their Pan-genome work that they've been doing in building ref graphs over time. So I guess you can take it away, Karen.
Thank you so much for that introduction. Hopefully everyone can hear me. Wonderful. So my name is Karen Miga. I am an assistant professor at UC Santa Cruz and an associate director at the Genomics Institute. I'm also the co-founder of the Telomere-to-Telomere Consortium and the director of the Genome Center for the Pan-genome Reference Consortium. And today, I think it's going to be a lot of fun. I wanted to walk you through some of the things that we're finding in terms of new genome biology and T2T genomes and where that's leading to the Pan-genome project, which is where my colleague Benedict Paten will take it and have a next series after that. So the human reference genome, as we all know, has been pretty foundational for genetics and genomics. It's been the driving force or the singular coordinate-based system that we've all been using to annotate new genes, clinical genomics, and understanding human histories. Of course, there's a challenge here is that the original Human Genome Project was only reporting the euchromatic sequence. These are the parts of the genome that we know contain genes. And it was known even back in 2003 with this early release that there were still hundreds of gaps and millions of bases that had been missing from this original reference. We know now, even looking closely at these gaps, where they are. They're not randomly organized. They're in regions that we know are highly repetitive that are now marked with a series of Ns in HG38. There's roughly 200 million bases that are missing. And these mark areas of our genome that are important for genome biology, areas that contain gene families or duplications that are sometimes found embedded within chromosome arms in the subtelomeric regions, and regions next to the centromere are the areas that are important for proper chromosome segregation. And in fact, all 5 short arms of the acrocentric chromosomes, 13, 14, 15, 21, and 22, have been missing for 2 decades. So it's been a lot of fun celebrating the release of a first complete genome. This happened back in 2022 by the Telomere-to-Telomere Consortium with these faces I'm showing you on this slide where we all met for the first time face-to-face in Santa Cruz last year. And this was really resolved with a combination of 2 different long-read components. It was high coverage PacBio HiFi and ultra-long sequences from Oxford Nanopore, or a pore-based a sequencer, which is embedded in a membrane where you can take any length of DNA through the pore. Now, this new sequence that was issued back in 2022 was really wonderful in that it enabled new science. It wasn't just a series of A, T, Cs, and Gs. There was a tremendous amount of effort to go in and try to annotate new genes, look at detailed map of repeat classes, also looking at sites of transcription and modifications that are happening on this new sequence. All of that can be downloaded today. In doing so, we finally had a complete map of gene families that existed. What I'm showing you here is work from Mitchell Vogler and Evan Eichler from UW, where you can see in red all of the genes that had been included in this new complete reference that were missing from the original human genome reference, which is what most people are using. The vast majority of introduced sequences I'm showing in this pie chart, hopefully your eyes are jumping to the bright yellow, were tandem repeat classes known as satellite DNAs. These are typically found in heterochromatic or densely packaged regions of the genome, such as centromeres. And if you zoom in, like on the cartoon that I'm doing, you can see these tandem repeats are a series of sequences that are found in a head-to-tail orientation and are typically riddled with repeats. Not only tandem repeats, but you have segmental duplications and transposable elements because it's really a more complicated region of the genome just to get right. And so in this complete genome, we finally have high resolution maps of all of these highly complex tandem repeat regions that I'm showing you a cartoon here. And this marks every single centromere of a human genome. And so this has been really landmark for many studies that have gone on from there. We've also gone through and looked for new sequences that had never been seen before. And in doing so, we're finding tandem repeat classes that had never been described. Here I'm showing you two in particular. And then you can see on the chromosome ideogram, the sites where we find all of these different classes showing up. So it's not just one place. These are classes that have been routinely missed because we've had an incomplete genome. And more recently, the telomere-to-telomere consortium took on one of the biggest challenges, and that's the Y chromosome. And in this case, I'm really illustrating that roughly half of the chromosome that's been missing is that beautiful kind of patchwork-looking quilt, which represents a whole nested group of tandem repeats that had been missing previously from prior studies. So these complete telomere-to-telomere assemblies have really been changing the way that we do genetics and genomics and the standards of what we consider to be high quality. And this has a ripple effect into how we can think about more comprehensive variant studies with healthcare, how we can take these tools and begin to look at other T2T or complete genomes in non-human primates and ruminants and rodents. And it's been driving a lot of new studies to look at cell biology or the epigenetics in these regions. However, we also know that one single genome is still not enough. It can't possibly capture the global genetic diversity that we know exists in the human population. And because of that, it would increase genetic bias, and it will not benefit everyone to use just a single reference genome. This is really impactful when you start thinking about those repeats that we were just talking about, where we know the centromeric repeats, or the ones that we know are the tandem repeats, vary considerably as we start to look across individuals. Here I'm showing you one example from chromosome 2, where on the bottom you can hopefully see it's a tiny little triangle or a very small array, looking at the stained glass plot. And as you move up, you can gain sizes by, from 1 megabase all the way up to 3 megabases. This is the same thing for gene families, where we find in some individuals you may will have 1 or 2 copies of a gene, and others you may have as many as 6, and that could really have an impact on gene dosage and function in the cell. And it even scales to the whole chromosome arm. Those acrocentric short arms I was talking about before, we're starting to detect that they swap between different individuals. So you could have a different chromosome configuration without having a T2T genome. You would be unable to actually see these events. And finally, we know that one genome cannot possibly represent the human population because there are large events called structural variations that are very difficult to measure with short read data sets alone. Here, what I'm showing you in the cartoon is a 6 kb or 6,000 base pair insertion that's not found in the reference genome. And prior studies had shown that if you take short read data, you miss about 70% of these bigger events. This is a problem that has been in genomics, and we've known about it for many years. This is just showing GWAS, where once again, this is really emphasizing from the Martin et al. Paper in 2019 that the genomic data sets we have, and this extends to our reference genomes, do not match our known breakdown of our global population. So the whole windup to this is to really emphasize that the T2T consortium and the first complete genome are wonderful events. However, they were just a signal saying, hey, the technology's here. Why can't we use this technology now to expand and do more and more of these T2T genomes to better represent genetic and global diversity, as I'm showing in this cartoon here? So the next goal was really to break down the technological barrier or the wall that was in front of us to ensure that we could begin to scale these technologies. And that's when the telomere-to-telomere consortium formed a kind of a unique partnership with another consortium that both Benedict, Paten, and I represent, and that's the Human Pan-genome Reference Consortium. This was a call to action from our local NIH. It was with the goal to improve representation of genomic diversity. Here, our goal is to reach at least 350 diverse references or 700 phased haplotypes when we think about T2T. When we think about who's currently in the pan-genome that's being represented by the NIH effort, here we're focusing primarily on genomes that are properly consented for open access, unrestricted use. We're looking at only common haplotypes, or these are haplotypes that or allele frequencies that have a global frequency of 1%. Globally, we're looking at normal lines, and these are cell lines. We're really lucky that we've paired up with kind of a star-studded ELSI team for our next phase, which is to go into prospective recruitment in large urban areas in the United States, including the BioMe Biobank in New York. And this has really been big science at its best. This is really highlighting some of the triumphs of our Genome Center, where we've been dividing these long read production elements across several different production sites that specialize in hi-fi and ultra-long data, as well as these linked reads assays. And this, I love to include on the slide, the bottom, which are our company partners, which is really something that gives us an amazing advantage because we get to work kind of shoulder to shoulder with some of the most updated protocols. That's really crediting some of the more recent ones that have come on, where we've switched over to a couple of new technologies, such as the Revio, which is something that our team has been embraced with for about as soon as it came out, so the past few months, as well as we're starting to beta test now the duplex assay. And in doing so, we are constantly trying to evaluate these really highly accurate long read data sets that I believe will be part of this workshop. And so in total, what we're doing is this call to action of trying to identify a cohort of individuals that represent global genetic diversity, at least 350, and build these extremely high quality diploid T2T reference assemblies to go into the Pan-genome Reference Consortium. In doing so, we've taken on 2 automated diploid assembly steps. The first one that I believe will be introduced in your workshop will be Virco. This is software that was developed by Adam Phillippi's team at NIH. It takes in this hi-fi component, the ONT ultralong, and Hi-C. And it does it, and it builds kind of an initial string graph, which I'll talk about briefly, and then it takes the ONT to kind of resolve it into wider loops. And then we also use a secondary mechanism, which is called Hi-Fiism ultralong, or ONT, which is developed by Hung Lee's group. So just to kind of give a very brief overview of what Virco is doing, you're essentially utilizing these extremely highly accurate reads, such as hi-fi or PacBio, where you can start to build something called unitig. These are areas where the extremely high reads are identical to one another, and you can start to build a string graph where the unitigs represent sequences that are identical, and the edges represent places where they're connected. We then take the ultra-long component. This is typically, I have 60 to 70x. That's really on the higher end. We're typically in the range of 30x coverage, and we use it to thread through. And what I want you to see in this slide is that you go from a graph that has a lot of nodes or a lot of these smaller units to getting a fewer units. So in other words, you're trying to resolve these paths using the longer reads to thread across. You can then use Hi-C data or trio data to go through and identify the maternal or paternal path of these graphs. And then you can eventually phase them into maternal contigs or paternal contigs. Now, all of these assembly work that we do is all available, of course, through GitHub, and our teams are working to bring them into WDL workflows to make them available through cloud-based protocols. The T2T consortium recently has been celebrating the first release of the diploid assembly. This is the QV100 that is currently available to the public, where the goal is to take our team and to really emphasize the best assembly possible, trying to create an assembly that has one error every, 100 million bases. So it's been an incredibly push to get a near perfect, if not perfect, assembly. And why is this so important? There are lots of sites in our genome that are incredibly important for understanding medically important sites, such as spinal muscular atrophy, or SMA, that I'm showing you here. We know that SMA maps to a region on chromosome 5q13, which is represented by a complex site with a segmental duplication or paralogs SMN1 and SMN2. And these genes sometimes have crossing over and conversion and have variable numbers that could actually contribute to this disease. If we look at just hi-fiism alone, or only using hi-fi data with either Hi-C or trio phasing, we find that often there are breaks in the assembly at this important site that would be important for us to get right for the medical community. However, with the newer methods that include both hi-fi and ONT, we're able to fully resolve these medically important sites, and this is really important for our reference genome effort. And so by taking toolkits like I'm showing here, developed by Jason Chin, where we have the Pan-genome reference, our research toolkit, we can begin to study the organization of this SMA locus, even as you go through pedigrees. Here I'm showing you 3-generational pedigree. We can begin to look at the complex organization across. And so once again, it's taking in this population sampling, building the T2T genomes, but the next thing that's really important is how you take these resulting T2T assemblies, form a multiple alignment, and then report something that's a reference resource. And here we're talking about a sequence graph, and this is really something that I wanted to pass the baton, so to speak, to my colleague Benedict Paten, because we've been celebrating recently the first draft human pan-genome reference. And this is something that I'm hoping that he'll be able to describe in more detail. Thank you very much for your attention, and I'll be happy to take questions at the end.
Thank you, Karen, for a lovely presentation. I don't know if we can allow Benedict to present his to do his presentation, then we can follow up with questions for both presentations.
I don't mind either way. If there are quick questions now, that's completely fine. Otherwise, I could just go.
Okay, no problem. Are there any questions from the attendees? Yes, Melissa, I can ask.
Thank you for the lovely talk. I just wanted to know, because I saw the bottom of Africa was sort of not represented in the one slide, but do you have any Southern African individuals represented?
So our cohort that we've been working with has been exclusively based on the 1,000 genomes collection. And the initial draft pan-genome only used a very small subset. It was the first 30, where we had originally kind of stacked our production in a way that wasn't very meaningful in terms of trying to front load a lot of the representation that we have from Africa. But we hope to expand into more 1,000 genomes efforts that are representative in Africa in the future.
Any other questions? Let me just check quick. Hossein, you can ask.
Thank you for this presentation. I just wondered, when you say highly accurate, is it you comparing it to the reference genome or to something else? I don't know. Thanks.
Right. So we have a number of, and hopefully this will be part of the workshop, to QC these particular assemblies. We have a number of different QC protocols that we didn't go over today. Ones where we take Kamer sets, for example, of Illumina or hi-fi data sets where we have an expectation, and we go into the assemblies to try to do QCs. This is using things like YAC and Marrow. We also align all of our read data sets back to the reference assemblies and look for coverage errors or errors that we might find in allele frequencies. You'd imagine that if you're looking at a haplotype phase and you're aligning reads to it, you're only expecting to find one allele over every single base. And so we do these types of screens using software like NukeFreak as well as Flagger. And so we've developed a whole panel of QC software to really go through and look at this without having to rely on the reference genome. As you could imagine, that could introduce biases.
Thank you. Thank you.
Are there any other questions from attendees? Let me just quickly check the chat box if there are any other. Well, there are no questions. Thank you, Karen, for your wonderful presentation. I hope the attendees enjoyed it. I did. Our next speaker is Dr. Benedict Paten, who will also be presenting exploring these studies of global genomic diversity with complete telomere-to-telomere assemblies. Sorry. You can take it away, Dr. Benedict.
Yeah, hi. Hold on, let me just flip back. Yeah, so I'm Benedict. I'm an associate professor here at UC Santa Cruz. So it's my pleasure to talk to you today. And I'm going to kind of follow on from a lot of the things that Karen was talking about and focus on some of the downstream work that comes once you've been able to generate some of these assemblies, and to think about what you can do with them. Just because of the question that was just asked, I just popped this slide in. So for the initial draft pan-genome that we created, we have 47 samples in the pan-genome, and you can see the sampling of genomes. These all came from the 1,000 Genomes project because the data was available to us. and it, was already consented for our purposes so that we could release the data. And you can see that 1,000 Genomes had a number of different sampling sites from across the world. And we essentially picked samples to attempt to maximize the effective coverage of that diversity. But of course, 1,000 Genomes didn't sample every single possible human subpopulation. And so there are definitely gaps in our current sampling. For example, there are gaps in the Middle East. We know there are gaps in Africa where there's a rich diversity, and that is something that we hope to complete over the next several years, working with partners, as Karen described. So so taking it from what Karen was talking about, if the cartoon picture of our project is that we have individuals who have contributed their genome data, and each of their genomes can be thought of as a pair of sequences or a pair of haplotypes, representing the maternal and paternal contribution. Then when we take those assemblies on their own, of course, they're all very similar to each other, and it isn't clear how you relate them together. So in order to relate them together, to understand which bits of the assembly represent the same sequence and which bits represent divergence, we need to align those assemblies together. We need to compare them. And to do that, as I'll talk about, we've pioneered several different approaches, but the net result is the construction of an alignment that we represent using a graph structure that represents essentially the parts of the sequence that are common and then the parts of the sequence that are polymorphic, that are different. So it's, I often get this question, and so I like to kind of talk about it right up front because it's a really important question. So people often ask me, why do I need to use a pan-genome alignment? Why can't I just use a population-specific reference? Why can't I just pick a genome in the set of genomes that we have assembled and just use that if it matches the sample that I want to study? Okay. And I want to disavow you of this notion and tell you why it's not a very good idea. And this is based on long-standing genetic observations. So we've known for a long time that most variants that you'll see in a genome are common variants, and they're mostly global. They're shared across all human populations, and they predate all of the divergence of modern human populations, right? Typically being about 1 million years old. And we know that most common variants actually don't vary in their frequency that much between most populations, with some notable exceptions, right? We can do this using things like FST estimates. They tell us how much do variant frequencies varies between populations, and we know that mostly they don't vary that much. And further, as I mentioned, most variants are actually common. So if you look in at any one genome, if I looked in your genome, I would see that 96% plus of the alleles in that genome are common. All right. The upshot of that is that any two randomly selected genomes from any significant population will differ by millions of variants, but a population-specific reference, so therefore a population-specific reference will generally only have a small effect, right? It's just because mostly any two people from any population will differ by millions of common variants, right? But in contrast, a pan-genome of just a few hundred individuals will represent the large majority of alleles in any sample. That is, we don't need to build a very big pan-genome to get almost all of the common variants. Okay, so that's an explanation for why it's really important to have a pan-genome, because we need to be able to represent the variants that are shared in our population properly. So now I'm just going to talk a little bit about how we built up the pan-genome and the things we can do with it. So, as I said, we represent the pan-genome alignment as a graph, because a graph allows us to view any kind of of genetic variation in a structured way. So in our graph, it's like a traditional mathematical graph. We have nodes and we have edges. So the nodes in our representation have sequences assigned to them. So they're like little snippets of DNA. And the edges connect those snippets of DNA, giving us the connectivity relationships or the topology of the variation. And then equally important is that we have paths that flow through the graph that represent the individual sequences or haplotypes. So in this picture, for example, we have a blue and an orange graph, an orange haplotype. that flow through our graph. And you can see that if you follow the sequence, for example, this blue sequence up here, and you spell out its letters, you'll see that it is reflected by this blue path that flows through the graph, and similarly for the orange. So one practical issue with building a big pan-genome alignment is that it's quite big, right? So for the draft pan-genome, we have about 270 gigabases of sequence, which is roughly 270 gigabytes of sequence, right, from just our 90 haploid genomes. And if we took them and we just said to people, okay, that's the pan-genome, you need to download that, then they'd be looking at, a really large file that becomes kind of impractical for the purposes of, doing bioinformatics. So of course you can compress that file. So if you use something like gzip, you can go from 270 gigabytes down to about 70 gigabytes. That's still a pretty big file. That's like a large BAM file, right? And of course, it doesn't represent any of the actual alignment relationships. So if you, instead of just, distributing the genomes individually as fast as, if you instead align the genomes together in a file called a GFA file, which is becoming the kind of standard way of representing pan-genome alignments, then because you're essentially collapsing stuff that's common, you go from about 270 gigabytes down to about 45 gigabytes, which is much more practical, but still pretty big. And you can, of course, compress that and get down to like 11 gigabytes. But in work from Yoni Sirin, who's a research scientist here at UCSC, he was able to create a new format that builds on the GFA format that takes advantage of the common features of the haplotypes as they flow through the graph to achieve even much smaller compression. So we can get it down to about 3 gigabytes, which is really pretty practical today for distributing over the internet. So this is the file format, if you will, that we plan and intend to use for most of our work. So actually, we've developed 3 different methods for building pan-genomes. Because this problem of building a pan-genome from a set of assemblies is kind of an open problem. So we have 3 separate toolkits, and actually more have come out since, but 3 that we trialed during the creation of the pan-genome project. So one, Minigraph, is from Heng Li's lab, and what it does is essentially generalize the Minimap2 aligner to building a pan-genome graph, and it's really, really fast. But one important limitation of it is that it's designed only to represent the larger structural variants. So it doesn't see the smaller variants in the graph. And then combining Minigraph with the Cactus tool from my lab, developed by Glenn Hickey and others at UCSC, we've developed this Minigraph Cactus toolkit, which essentially takes a Minigraph graph and then builds a detailed pan-genome, including all the small variants, right? One important feature of it is that it currently omits the centromeric variation. So it omits most of the satellite variation by clipping it out, just because we can't reliably align that sequence right now. And then finally, we have this pan-genome graph builder from Eric Garrison's lab, which constructs an all-to-all pairwise mapping. It has no notion of any reference, and it compresses and contains all of the sequence. But as a result, it is kind of larger and not so easy to map to. So in terms of the pan-genome, just a sort of broad strokes, if you imagine we've taken these 50 people and we've aligned them together, and now we're looking at the alignment and we're saying, okay, tell me about the different kinds of variation that you see. We see about 20 million small variants. So, obviously the majority of those are single nucleotide polymorphisms, so just single base substitutions. Then there's a smaller number of what we call multi-nucleotide polymorphisms, where a small number of bases that are adjacent get changed, that have apparently changed at the same time. And then, of course, indels, so small insertions and deletions. And just to orient you in these plots. So here we're looking at 2 different pan-genome alignment construction approaches. So both the Minigraph Cactus and the PGGB, and they both give us very similar results, indicating, as we showed in the paper, that actually there's a lot of concordance between the approaches in terms of how they represent the variation. Turning it around and looking at the larger structural variants, there are about 70,000 large structural variant sites in the draft pan-genome. And here we've just broken them down. This is a panel from the paper. We've just broken them down by different classes. So we've got, for example, the mobile elements up here, and then the tandem repeats, the VNTRs and STRs, and then down here we've got, for example, the segmental duplications. And the thing to bear in mind here is that many of the mobile elements are kind of biallelic, so it's a little bit difficult to see in the coloring here, but the dark coloring indicates that there are just 2 alleles at a given site. And if we look at things like ALUs, most of the time when we look at those structural variants in the genome, we only see 2 alleles. You've either got the ALU insertion or you don't, right? In contrast, when we look at the VNTRs, these variable number tandem repeats, and we look at those sites, we actually see that at any given VNTR site, yes, sometimes we only see 2 alleles, that's the dark colored bars, but often we'll see 3 alleles, and quite often we'll see many alleles, to the point that we will see a different allele for every single haplotype that's represented there. And that just indicates the much higher level of variation that exists in those sites, that indicates the high level of mutation that's taking place. We did a lot of work in the initial paper to validate the structural variation. I'm not going to go through that now, but just to say that we were able to show a high level of concordance between the variations that we discovered by our pan-genome alignments and existing knowledge of this kind of variation. And just to kind of give you an illustration of what the pan-genome looks like, in much of the pan-genome, you can imagine it looking like a traditional multiple sequence alignment. So if you've ever aligned together any set of sequences, you'll get back an alignment which looks like a series of rows where the columns represent bases that are aligned together, interspersed with gaps. And most of the pan-genome can be represented that way because mostly that's that's how our sequences are. That's how the changes have arisen. But in maybe 600 sites in the genome, there are these larger common variations that exist that make quite complicated structures. So here I'm showing you the HLA-A locus, which is part of the major histocompatibility complex, the MHC, that's involved in antigen presentation. And so in HLA-A, there are several paralogs of this HLA-A gene that we've got labeled here. So this is showing you the graph. So imagine, we've zoomed in on one small piece of the pan-genome and we're looking at that specific place. And if you will, this is kind of a lobe of the pan-genome. And we've colored the individual copies of the gene by these sort of green to blue colors, right? And you'll see here that, well, one thing at a high level, it's definitely not linear. There are kind of, clearly loops and structures that would not be represented by a traditional multiple sequence alignment. And I want to draw your attention to this HLA-Y allele. So it's been known for a long time that HLA-Y is an additional paralog, an additional gene that's present in some people, but we didn't up till recently know where this gene actually was, where it fell on the sequence. Well, with the pan-genome, so here's the same graph as before, and now I'm showing you different sequences that flow through the graph. So for example, here with GRCH38, you can see that GRCH38 flows through the graph this way. So this is the current reference, I should have said. So GRCH38 is the current reference sequence that you would use if you were mapping to a genome. And you'll see that it's missing this HLA-Y sort of loop, right? But actually, here's another sequence from the pan-genome, and you can see it flows through the graph, going from dark red through to sort of purple, right? And you'll see that it does include HLA-Y. And in fact, when we looked at it across our samples, about 28%, so roughly a third of our samples have this, or a third of our haplotypes have this HLA-Y loop. So now, for the first time, we can say where HLA-Y is. And of course, with this alignment, we can kind of orient it with respect to our existing reference and really have a richer appreciation for the complicated structural variation that exists at this loci. And in the paper, we went through a number of different examples of these complicated structural loci. Karen showed a really nice example with the SMA locus, for example, which is a pretty challenging loci, actually. And I think in the future, getting the pan-genome to represent these difficult regions is a really critical task for us as a community. Okay, so in the time I have left, I want to briefly talk about applications of the pan-genome. Because it's one thing to go and assemble sequences and then, of course, build alignments. But it's another thing to then go and make make the alignments, make the pan-genome actually useful for people. So we previously developed this mapping tool for mapping short reads to a pan-genome reference. We call it VG Giraffe. So VG is this toolkit that I think Glenn may be talking to you about, that essentially represents a kind of pangenomics toolkit. And within VG, we have this tool Giraffe that is this mapper. And the short way of explaining it is that it's a very fast mapper that relies on the fact that once the pan-genome represents most of the common variation, mapping a read into the pan-genome becomes an exact matching problem, where you're essentially, without any gaps, trying to find a gapless alignment of that read to a location in the pan-genome. And because you don't have to worry about gaps in the alignment process, the whole process becomes very, very fast. So as part of the Pan-genome paper, we developed this pipeline, which is available for anybody to use, that takes Giraffe and then combines it with the Google Health Deep Variant software, which is a variant calling software. So what we're effectively doing is taking a bunch of reads, mapping them to the Pan-genome with VG Giraffe, and then using deep variant to call variants given the mappings. But an important limitation of the way this works currently is that Giraffe maps to the full pan-genome, but because deep variant only understands a linear reference, what we actually do is take those initial read mappings and then project them back onto a linear reference from which we then run the variant calling software. And without sort of dragging you through the details of the results, this is a precision recall plot, but it shows that, for example, when you're using this Giraffe deep variant pipeline, actually with trio data, you can get really, really, really good performance that is much better than, for example, what you would achieve with a linear reference. Another really important application of the pan-genome, so that's sort of focused on the VG Giraffe deep variant workflow is currently focused on small variants. Another really important class of variant, obviously, is the structural variants. And we developed, this is work from Jana Ebler and Tobias Marshall's lab, developed this tool called Pan-Genie, which essentially takes the pan-genome, uses k-mers from a given sample to guess the structural haplotypes of a sample across the genome. And using that approach, we can actually double the number of structural variants that we can find. So we can go from the minority of structural variants that are discovered in a given sample. So here in green, this is on the y-axis, we have the number of structural variant alleles that are being detected in a sample. So this green represents what you can do if you're essentially using standard tools mapping against the linear reference. And the blue represents using Pan-Genie, using the Giraffe pan-genome. You can see that we effectively double the number of structural variants that are discoverable. You can read more about this in their paper. But we think this is a really important, task for the pan-genome that really improves our ability to go and assay these kinds of variants that have really been missing up to now in many of our kind of association studies, just because they're difficult to discover. I won't take you through this, but just as a sort of advert, we've also developed RNA-seq pipelines for mapping and quantifying expression of transcripts using the pan-genome. And if you're interested in this, we had a paper that came out on this earlier in the year, and we can show that pan-genome mappings to, RNA-seq pan-genome mappings are more accurate than those mapped against a linear reference. And the upshot is that we can get more accurate expression quantification. And then the last thing I wanted to talk about is just where we're kind of going over the next, let's say, couple of years. So our pan-genomes are going to grow. We're going to go from 47 genomes to 350 to then 500 over the next several years, and probably larger than that, right? So actually, if you think about it, as the pan-genome grows, each new genome that we add is actually going to add mostly rarer variation as the sort of tail progresses. And that's just because, once you've added, 100 genomes, then the chances that a new variant that you're adding in the 101st genome is is kind of common, it gets quite small, right? Because the chances are you've already seen the common stuff, okay? And the issue with this, I mean, it's great that we can grow the pan-genome, we can represent more diversity, but the issue is that as we grow the pan-genome, that rarer variation actually starts to act a bit like noise, right? So if you're looking at a new sample and you're saying, okay, how does my new sample relate? You've got to suddenly contend with, you know, lots and lots and lots of additional rarer variations that are not relevant to your sample, right? And that can actually, in, for example, read mapping algorithms, cause mismapping. So the way to, you know, easy way to visualize this is to consider this picture on the right-hand side here. So this is zooming right into just like one, a very small site of variation in the pan-genome. And you can see in the pan-genome, there's a SNP. So there's either a G or a C, and there's also it's kind of a complicated site. There's a potential deletion of that one base as well. So there are 3 possible alleles. You either have a G, a C, or you skip the base. And you can see the reads, which are shown here as these little red ribbons. They either include the C allele or they skip the base. Apparently there's one read that skips the base, right? But if you think about it, that G allele is not present in the reads. So this, in sort of gray, I guess, are the different haplotypes that flow through this. And there's only one haplotype, which I colored in purple, that contains this G allele. Now, if you think about it, across the whole genome, as we add more and more and more variation, we're going to have more and more of these examples where we have these additional alleles that potentially, if there are errors in the read, can attract the reads to map to them and then essentially cause ambiguity about where those reads should map. So in the long run, as pan-genomes get bigger, it gets harder and harder to actually know where to map the reads correctly. So to date, to solve this problem, what we've been doing essentially is saying, okay, well, we have this rich pan-genome with this additional variation in it, but we're going to essentially ignore the rarer stuff for the purposes of, for example, read mapping. So I won't go through this, but we've implemented a number of different heuristics, which basically just use allele frequency to filter variants that are rarer than a certain point, just because we know that at a certain level, they just create noise, right? But that's obviously a huge waste, because in any sample that you're studying, there will, of course, be many thousands of rarer variants. And so it's really important in the long run that we can utilize that information. So just, again, I'm running out of time here, but we have been recently working on methods that essentially use this method of what you could consider local ancestry inference. So every location in the pan-genome, if you imagine scanning along the pan-genome, we are picking haplotypes that at a given location are relevant to the sample that we're studying. So imagine, we've got 100 haplotypes that we could pick from, and we pick 4 or 8 haplotypes. And in fact, having picked, say, the 8 haplotypes that we're closest to, we could then actually pick the best diploid pair, because obviously we think that our actual sample is diploid if we want. So we can get all the way down to a pan-genome subgraph that represents just our best pair of haplotypes across the genome that we think best represent our sample. And we have that now working, and it's very fast. So here's how long it takes to map BWA-MEM to map a set of reads to GRCH38. And here's how long it takes to map using that diploid sampling to the pan-genome, including the process of building that personalized pan-genome. And just to very briefly say that the results are really, really good. They are really state-of-the-art, substantially better than you could achieve with, for example, the genome analysis toolkit, the GATK. We have far fewer errors when we use this approach of creating a personalized pan-genome and mapping to that for the purposes of our pipelines. And similarly with structural variants, we're starting to kind of approach the performance of long read methods, just using short reads when mapping and then variant calling against the pan-genome. Okay, I think this summary slide, sorry, I just pulled this together yesterday. So I just want to kind of emphasize to everybody that the pan-genome that we're building is something that we as a genomics community can all use to equally represent, or as far as possible, equally represent our population. The pan-genome that we've released thus far adds about 120 megabases of new sequence to the human reference in euchromatic regions, much, much more in the heterochromatic sequence. It's a huge amount of assembled sequence at really high quality. I haven't talked about this, but it represents many gene duplications. And of course, millions of small variants, 70,000 or so structural variants. And the last point I think is important. We know, Karen and I know, and others who are working in this space know that the adoption of the pan-genome is going to take time. And building it completely is going to take time. And while I think the things that we've been developing are quite promising and useful right now for different kinds of tasks, to actually get broad adoption, we might be talking about 5 or 10 years over the till we're at that point in time. So I just this is a plea to people to be patient and to try out these things out and help us get to where we're trying to go, which is hopefully a better future for genomics in terms of our reference. And yeah, I just want to acknowledge the folks in the lab. And yeah, I'll stop there.
Thank you, Benedict, for your nice presentation. I don't know if there are any questions from the attendees. Sorry.
That is totally fine.
Yes, Arash.
Hi, thanks so much for a nice presentation. So my question is about the pan-genome construction at the initial steps. So I want to know how variants are detected. They are called based on the multiple sequence alignment of assembled genome, or they are directly called from the short reads or any kind of sequencing file read.
That's a great question. So yeah, we did it both ways. So Ira Hall's lab took standard reference-based approach where they mapped the reads to GRCH38 and they called variants. And so we created VCF files for both small variants and structural variants using the sort of, if you will, standard reference guided approach. We also, as you alluded to, created the multiple sequence alignment, right? Which, if you think about it, like I said, I was just showing you pictures of like bubbles, right? It sort of represents places where sequences diverge. We then projected those differences back onto a reference and created VCF files. And that way we had, two representations of the same thing derived from completely different means, and we could compare them. And we showed that they were really, really similar. Does that answer the question?
Yeah, cool. Thank you so much.
Yeah.
Any other question?
What's the question?
Okay, I guess we can start with the question in the chat, and then Melissa will respond to your question after. So,
I'm getting an echo.
No, you can proceed. You can proceed.
Okay, sorry. So how many genomes might be needed to have a more complete pan-genome? That's a great question. It's something that we've obviously been thinking about a lot. Karen, you might want to take that one actually in some ways, because I think it's more of a population sampling group. I mean, I have an opinion, but
I think there's a lot of unknowns. We're only able to really survey common variation where we have databases. And so, for example, when we look at the 1,000 genomes database, we have a VCF file of all the common alleles. And we know that when we apply some of our greedy algorithms to look at the coverage of these common alleles, we have saturated that particular database where we know we're representing over 99.9% of the common allele space. We're taking that to the Nomad databases and Biomed and starting to look outside of the 1,000 genomes. And when we perform this over exome or over limited space, we're finding that we still have about 2%, of variation to go. And what that kind of was telling our team early on is that these very small variants that are easy to measure with either microarray or, that we really have a nice coverage right now with a very small number. And that's echoing what Benedict said in his presentation, that we expect that type of sampling of common or older haplotypes to not demand such a higher number. However, as Benedict also mentioned, there's a lot of dynamic regions that we know are complex, and these could involve medically relevant genes that are gene families or, segmental duplications or satellites. And those we do think we will need a much higher number. And so in that case, we're starting to incorporate more long read mapping and looking into the allele frequencies in these areas. And there's also a lot of interest in expanding just in general, outside of Nomad, outside of Biomi, to start to look in some of these regions that Benedict indicated on his first slide. We don't have a good census right now of allele spectrum, and that we're probably underrepresenting. And I'll end this, I'm sorry, it's a long-winded answer. I think the overall goal of the pan-genome is just kind of benefit driven. So no matter who walks into a clinic, no matter in the world, they will have representation in the pan-genome, that will allow them to have some benefit. In other words, just some meioses back, they'll have representation of a haplotype. And so over time, maybe this will take 5 or 10 years. This will be a very iterative process, but it'll be to ensure that that type of representation of individuals who are extant now that are around the world could actually find benefit with their respective haplotype will be the ultimate goal. And that number, I think, is not known, but it probably will be in the range of thousands instead of hundreds.
Thank you, Miga. Melissa, you can go ahead and ask your question.
Thank you for the really wonderful talk. I just wanted to ask a question about your slides on the personalized pan-genomes, just to understand that a bit better. So is that at the level of global ancestry for an individual? So saying sort of inferring what their best represented haplotypes would be, just because I work a lot with admixed individuals, or is it at the kind of local ancestry level that one is talking about?
Yes, so it's a great question. I rushed through that a little bit. Sorry, I sort of shoved together a bunch of slides kind of quickly. So very understandable question and good question. So we're essentially at each location in the genome estimating the local ancestry and picking haplotypes in the pan-genome that represent the sample that we're interested in, right, at that location. But of course, that implies, we might pick different haplotypes at, let's say, the ith location as we pick at the i+1 location, right? So as we're scanning along, we're picking slightly different haplotypes. So in order to allow that, we have to recombine, in silico, the haplotypes that we're picking. So we basically construct a sort of artificial set of haplotypes that at each location represent our individual that we're studying as best we can, given the samples that we have, right? But that are not actually any one of the genomes from our set. So let's say that we took your admixed, take an admixed sample that we're studying. And if we have representation of those haplotypes in our pan-genome, we can, in silico infer an admixed representative set of haplotypes that will closely represent our sample. So this should work well for admixed individuals as it works for anybody from any population. Does that answer the question? So it's definitely local ancestry inference as opposed to trying to kind of globally say we're going to pick, I don't know, this genome N and genome J from our set to represent the individual, because you're right that, like, as I was trying to say at the beginning, in general, you won't be able to find, any pair of genomes in our pan-genome that will adequately represent one person.
Thanks very much.
Benedicta, we have a question in the chat box from Paten.
Sure, thanks. So asks, does T2T sequencing require samples with more stringent quantity quality requirements, or are samples collected for traditional 30x whole genome likely to be adequate? I think, again, Karen, you should probably take this one, but I'm happy to.
Yeah, no, this is really kind of harkening back to our long read production centers as part of the pan-genome effort. And we start off, in order to get these extremely long reads, with a lot of starting material. And we need this starting material in terms of cell pellets or isolated DNA to have intact high molecular weight DNA. And because of this requirement, I think that's quite different from 30x whole genome shotgun. And so we've been or sequencing. I think that's something that we've been taking quite seriously. The hi-fi components are a little bit more stable, but you still need a large amount of starting material on the order of like tens of millions of cells in order to make these protocols currently work.
I also see get an echo again. Oh,
Yes, there's a follow-up question from Mamana in the chat box.
Thank you. Yeah. Do you think ancestry population-specific pangenome would be useful? Ancestry population. So that's an interesting question. So people have been experimenting with constructing sort of pangenomes for just subsets of samples. So, for example, there was recently a Chinese pangenome that was a draft that was published in Nature that took a bunch of individuals of Chinese. I don't know, it's a complex answer. And well, geography, I should say, and constructed a pangenome. And similarly, I know some people have been constructing, for example, an African pangenome. So I think that people are going to experiment in this way. Our approach within the HPRC, and I think this is consistent with the science that the genetics, is that actually a pangenome built from people across the world will best represent will be adequate to represent all people, and that actually population-specific pangenomes, whilst they may be interesting and useful for purposes of studying kind of ancestry, will not necessarily be better for actually representing any given individual.
I don't know if there are any other questions that oh, so there's a question from Mohammed in the chat box again.
Yeah, so Mohammed asks, regarding haplotype-aware pan-transcriptome analysis, do you have a published workflow for that? So within VG, there are tools that are public and available for doing that. And some people have been using those. We don't have a single workflow like on Terra to do that, as far as I'm aware. But we definitely do have tutorials that we can probably point you to if you're interested. So send me an email and I can direct you. Karen, there's a question, I think, for you.
There's a question in chat once again from Melissa. Thank you, Melissa. Can researchers contribute sequencing data to the global pan-genome effort? I think one of the criteria that we currently have for the pan-genome is that all of the data that we're utilizing needs to be properly consented for open access data sharing and use. And other than that, I think that's been kind of something that we've been trying to work with international teams to ensure that we do have those kind of shared consent structures. And that's working through GA4GH. So we've recently launched through a driver project with GA4GH, the Human Pan-Genome Project, which is a global effort to create this pan-genome, which has kind of investment and shared resources across many different researchers around the world who are contributing, as you mentioned, sequencing data, but with this idea that it should be open access. There's
I see. Somebody asked, how accurate was GSCH38 for the pan-genome? I mean, so we're all big fans of GSCH38. It's extremely useful reference that has, which is very high quality for many regions of the genome. But of course, it is missing certain regions, as Karen was talking about earlier. With the T2T, we were able to show the 200 megabases of sequence that was just either wrong or gapped or just simulated. So GSCH38 is a really useful reference, and we definitely view it as being an important set of coordinates that we're going to include in our pan-genome. So for as far as we can, people will always be able to use GSCH38 to talk about locations in the pan-genome. But there are some places in GSCH38 that just don't represent human sequence adequately. And for that, we need other references. And so, for example, CHM13, but others in our pan-genome as well will be better representations. So we see the pan-genome building on references like GSCH38 to create a more comprehensive set of representation. I see a question about adding to the pan-genome. So do we have to rebuild the graph from scratch, or can we update it? So that's a really interesting question. The mini-graph algorithm does allow incremental addition. In fact, that's kind of how it's designed. So, you start from a reference and then you add one sample on, and that builds a sort of slightly more complicated reference, and then you just repeat that process, adding new samples in progressively. So you can definitely use mini-graph that way. With the downstream processing, because of the way we build the multiple sequence alignment, it's quite difficult to do that in an incremental way. You kind of have to consider the relationships between all of the sequences in order to construct the multiple sequence alignment accurately. But in principle, it would be possible to develop a tool to allow you to do that using some alignment algorithms. But currently, maybe Glenn wants to comment, but currently that's not a mode that we currently support. Hope I got that right, Glenn.
Right, yeah, only mini-graph, not mini-graph cactus. I don't think PGGB has an update either.
Right, exactly.
Something everyone asks about. Hopefully we come up with something soon.
Yeah.
Well, if there are no other questions, I really would like to thank everyone again for attending the seminar. It's been wonderful. These are very thoughtful questions. Thank you for your attention and attendance.
Yeah, thank you so much.